In Chapter 5, we're going to be taking a look at a variety of different kinds of eukaryotic microbes. And if you take a look at this figure here that I got from a different textbook, I just pulled this out to show you how this idea of microbes that fit in the eukaryotic groups have changed over the past few centuries. And if you go back to Linnaeus's original taxonomic groupings that are out there. For him, everything that was living was grouped as either belonging to kingdom Animalia or kingdom Plantae. You either moved and ate your food, or you sat there and were able to produce your own food because you could do photosynthesis. And you can see by the time Whitaker came up with his taxonomic system in the 1960s that he proposed, we have the addition of this kingdom protista that happened earlier, actually, in the late 1800s there. And his major contribution was to propose that these organisms that were grouped with the plants out there that turned out not to actually be plants should be grouped together in this kingdom fungi. Nowadays, if we look at this, really, you're talking about 50 years after Whitaker's proposals here, people started to realize that this other kingdom, Protista here, actually looks like it has a whole bunch of different groups of organisms in. So they're looking at taking this kingdom Protista and actually breaking it up into a bunch of different kingdoms there. So just as with living organisms out there, when we look at the classification of these organisms, that is subject to change as it evolves. If we look at this sort of modern idea of phylogeny and phylogenetic trees that are out there, we can see this common eukaryotic ancestor is right here at the root of the tree. And then you can see these branches that have occurred and all the other branches that give us the modern day of what phylogeneticists will call supergroups of organisms that are out there. And within these supergroups that we can see listed over here on the right side of this figure, we've got our various kingdoms of life that are out there. And some of these, you can see this little dotted line right here. This is a relationship that is suggested by the evidence that is out there. And so that's really something that is still under debate. And so we'll take a look at a variety of the different characteristics for these groups of different kinds of eukaryotic organisms. And just to point out, that's us way down here in the animal kingdom. And you can see there's a lot of different kinds of groups of these organisms that are eukaryotic, but they don't fit into the fungus kingdom, the plant kingdom, or the animal kingdom. So that's why we have all these other proposed kingdoms that are out there. If you take a look in the book, there are a couple of tables there that show you some of the different characteristics of these different groups. So you can see the supergroups that are listed right here that were shown in that previous figure on the extreme right side there with your supergroups. And then within there you have these subgroups. So this is where, depending on which kind of system you're looking at, you could put this as a kingdom or you could put this as a phylum. And again, it depends on who's doing the organization for that taxonomic system there. But you can see some of the features of these have been elucidated in this table, along with some of the clinical notes here on different kinds of organisms and the diseases that they cause. And just sort of as a reminder, when we look at the eukaryotic kingdom, remember we're looking at organisms that are primarily characterized because they do have those membrane-bound organelles inside of the cell. So the things that we saw in Chapter 3, like the nucleus, you've got the folds in the nuclear envelope that make up the endoplasmic reticulum. You've got a variety of different features that you can find floating around inside the cytoplasm of the cells, depending on what you're looking at. You can find things like mitochondria and chloroplast. You can find vesicles or sacs that are made up of membranes that are used for transport and storage. You can also find things like the Golgi body that's going to be involved in processing some of the chemicals and then shipping them around in the cell using those vesicles. The other major difference when we look at eukaryotic cells is the idea of reproduction or cell division. And when we look at cell division in eukaryotes, remember that you can do two different versions of it. You can do mitosis, like is shown in this figure here, where the cell is basically duplicating its DNA during interphase, and then when they enter cell division by mitosis, they go through those characteristic kinds of phases, which can be seen in the microscope when you're looking at the chromosomes as they're being moved around within the cells there. So I'm not concerned about the phases of mitosis here, but I do want you to remember that when we look at this process here, if you start out with a cell that's got two copies of all the chromosomes, known as a diploid cell, at the end of this process here, you're going to end up with the same type of cell there. So here we've still got our two copies of our chromosomes in the nucleus of the daughter cells there. So the cell is literally going to be maintaining itself or cloning itself. So not only are the daughter cells genetically identical to the mother cell, but you still have the same number of chromosomes as a result of mitosis. When you look at this process of mitosis, it can be used to reproduce, and when you look at multicellular organisms like plants and animals, it's going to be used to produce new cells so that you can grow and also to maintain that growth and replace dead or dying cells. When we look at the other version of cell division, when we look at meiosis, remember with meiosis, you go through two phases of division that are reminiscent of what you see with mitosis, with some distinct differences there. During the first part of meiosis, remember that's where you can have homologous chromosomes, so the chromosomes with the same array of genes are going to do crossing over, so you are literally breaking strands of DNA and then alternating them or switching them between the chromosomes, and that's going to result in genetically distinct daughter cells coming out of this process, as opposed to mitosis where you're getting clones. And the other thing is, because of these two successive divisions here, we're going to have a cell that starts out with two copies of the DNA, ending up producing four cells with a single copy, because remember that two copies of DNA is copied during the process of interphase. So you have a double diploid cell effectively here when we're looking at this process. And then at the end of this process, you end up with four haploid cells. And remember, when we look at this process, we always think of it for sexual reproduction. But we'll see when we look even in the fungus kingdom in the next section here that they actually exist in the haploid state for the majority of their lifespan, and the only time they are diploid is when they're going through the sexual reproduction process. So these characteristics of cell division in eukaryotic cells are going to be a lot more complex than what we see with our prokaryotic cells, where you're basically duplicating your DNA and then the cell just gets bigger and divides in half. Here we have to maintain the chromosome numbers, and when you look at something like a human, we've got 23 pairs of chromosomes, so a total of 46 that have to be maintained so that you end up with your 23 chromosomes in each of these haploid cells there. So a lot more complex kind of process when we look at the eukaryotic microbes that are out there. We'll start out looking at the fungus kingdom, and the fungus kingdom's kind of interesting because it turns out when you look at this phylogenetic tree, notice there's the animals. We are more closely related to fungi than we are to a lot of these other kinds of eukaryotic organisms that do have animal-like characteristics. When you look at the fungus kingdom and you look at their nutrition, they're what's known as heterotrophs, and they get their carbon from their food. So they are like us in that sense. They are different because they are known as saprobes, and that just means that they're decomposers. And they're basically going to be absorbing or ingesting small food molecules that are floating around. And the way a fungus eats is kind of similar to a lot of the bacteria. You can release some digestive enzymes into the surrounding area and decompose your food so that it's smaller and smaller, and then you can allow that smaller food to get across your membrane and into the cell to provide that carbon and energy source for them. When we look at the fungus kingdom, the other big difference here is they do have chitin that makes up their cell walls. And when you look at chitin, it is made of NAG molecules. So that one of those sugars that you have for peptidoglycan, NAG and NAM are the two sugars. They're going to basically co-opt one of those sugars, the NAG molecules, in acetylglucosamine, and then use that to make these long chains. But they don't have those peptide cross bridges like we see with our prokaryotic cells that are using peptidoglycan. And the other major difference that we see when we look at the fungus kingdom out there is their membranes, like other eukaryotic cells, do have sterol molecules. But where animals use cholesterol, fungi are going to use something known as ergosterol. So they do still have those sterol molecules that are embedded in that fatty acid layer in their membrane, but it's a different version of it. And this is actually useful because remember, when we look at the characteristics that all cells have, you have to have a membrane, you have to have DNA, you have to have ribosomes and a cytoplasm. If we can interfere in any one of those four features, we could result in the death of cells. So knowing that there's a difference in the sterol in the membranes for a fungus, that means that we can look for antibiotics that interfere with that sterol, which could interfere with the membrane, and now you've got an antifungal target to try and get rid of the fungal infection while still allowing the animal cells to survive that process that's going on. When we look in the fungus kingdom, we do find there are a variety of different versions of them. There are microscopic fungi that are out there that are yeast cells, and here you can see they're kind of an oval or a spherical type of cell. This is not a coccus like we see with the bacteria. And those will actually grow up, as we've seen in lab, kind of like bacteria. And when we look at the macroscopic versions of the fungi that are out there, these are the molds and the fleshy fungi. So think mushrooms. And when you look at them under the microscope, you will see these long filamentous thread-like cells that are out there. And so distinctly different kind of feature, but they still share those characteristics. They're decomposers that have a cell wall with ergosterol in their membranes. And if you look at this sort of cartoon, you can see that these filamentous fungi are going to grow up to make a big structure. So all these long, thin Hypha cells that are out there are going to form this tangled mass. So think like a mold growing on the mushroom, or a mold growing on your orange out there, or a mushroom. It's going to make the body of the fungus that is known as a mycelium right here. And within this, as you look at the individual hypha that are out there, you can actually find some differences in the hypha structures that are out there. And so when we look at this, you can find some of these hyphae are going to make what's known as a septate hypha. And this one, if you look closely at this, you can see there are little cross walls between the cells, and the dot is the nucleus right there. So this hypha right here is going to grow by basically elongating at the tips right there. When we look at this one over here, notice these cross walls are missing, and so this is an aseptate hypha that is out there, or a cenocytic hypha. And this is something that we'll see when we look at the slime molds later on. They're just lacking a cell wall around here for the plasmodial slime molds. And this one over here shows you how yeast cells can actually sort of stick together and make a long chain of these budding cells, and so it's known as a fake hypha or a pseudo hypha here. And I found this video put out there by the University of Massachusetts Amherst to show you some photo movies of some yeast cells, and here you can see what's known as cytoplasmic streaming. So the cytoplasm inside of these cells is literally streaming around inside of there, and you can see some of these organelles moving around. And so here you can see these two cells are still stuck together. And in this part of the movie, you can actually see the bud developing right there. So this is the process of mitosis that has gone on with these cells right here. And again, we can see that cytoplasmic streaming occurring in there. In this one, you can see how yeast cells can form that pseudo hypha. You can see these chains of cells that are developing here as they divide through the process of mitosis.